When microbes invade our bodies, they multiply and cause infections. In the past, antimicrobials could easily treat these infections by killing invading microbes. However, an alarming increase in antimicrobial resistant infections is rendering existing drugs useless in the fight against infection. The future of medicine depends on the discovery of novel therapeutics, and the answer lies in the microbe's DNA. Microbes exist everywhere in our world, and the tremendous microbial biodiversity present in the environment has yet to be fully explored. Complex interactions between microbes and their environment have resulted in a natural selection process where the surviving microbes have evolved to be stronger than their predecessors. This ability to survive is due in part to natural chemicals produced by microbes. Natural selection has evolved these chemicals to have distinct and privileged architecture. Scattered along the length of a microbial genome are biosynthetic gene clusters. Each cluster contains 2 or more genes which encode the information necessary for a microbe to build an enzymatic assembly line. An assembly line is responsible for synthesizing a natural chemical. An assembly line consists of several modules which pass a growing natural chemical linearly from one module to the next. The domains within each module dictate how the new chemistry is added. These chemicals can help microbes survive in nature. When interacting with competitors, the chemicals produced by one microbe can bind to a biological target in its competitor and cause a cellular change, sometimes leading to the death of the competitor. The microbial interactions in nature, aided by natural chemicals, are analogous to how drugs work in our own bodies to eliminate infectious microbes. They can provide, and have provided, inspiration for many pharmaceutical drugs. Penicillin is a natural chemical antibiotic that was discovered by chance. Many other commercially available drugs have been discovered in the same way. Repeated rediscovery of known compounds is common when relying on chance discovery. However, there are trillions and trillions of undiscovered chemicals in nature. So how do we stop finding the same chemicals and start discovering new ones? Genome sequencing is fast and inexpensive. The big data accumulating through massive sequencing efforts can be analyzed using informatics software. These programs scan the genetic code for biosynthetic gene clusters. The genes within these clusters encode the enzymatic assembly lines. The structures made by each of these assembly lines can be predicted. In the end, every natural chemical encoded by a genome can be identified. They can be classified into known chemicals and unknown chemicals. The chemical of interest can then be targeted for further exploration as a potential therapeutic. Informatic strategies that rapidly and efficiently analyze the big data available could be the solution to the antimicrobial resistance crisis.